So this slide does not have today's attendance password on it, but it will show up when we switch over to the next presentation. But if people need it, it's Photon. Again, you guys are pretty good at sharing that information with each other in the chat. So today we're going to pick up with where we left off on Monday. So looking at the structure of our polysaccharides. So just as a quick overview here, and I'm going to try to move your faces over there. We do have some structures here that I wanted to point out with regards to looking at starch and glycogen and cellulose. These tend to be the three main ones that we focus on, especially when we're looking at human biology in particular. There are different ones that we'll look at briefly for insects and bacteria that are important as well. But these are the three main ones that we tend to focus on. And what I want you to notice is that with both starch and glycogen, we're using those alpha glucose monomers. So remember, with the alpha, we have that nice hydroxyl group at carbon 1 that is, facing forward, as we called it on Monday. And so this is going to provide a certain structure as we start building these really large polysaccharides. So we're going to see that both starch and glycogen are fairly similar. They both use an alpha-1,4 glycosidic linkage. So that's going to build a nice long molecule for us. So a nice long polysaccharide. But in addition to that, they're also able to form an alpha-1,6. So if you remember this carbon up here, this is our carbon 6. And so they're going to be able to form a bond between this carbon and a carbon 1 on a different glucose molecule to allow for some branching. So this is going to allow branching in both starch and glycogen. And what we're going to see the biggest differences between starch and glycogen is how often that they're going to have those branches come off. So we can see that it's only about one every 30 glucose residues. We'll see it branch in starch, where it's a little bit more frequent in the glycogen. So we'll take a look at what that looks like here in just a second. Cellulose, on the other hand, is a beta glucose monomer of our glucose molecule. And so it's going to form beta-1,4. So again, we're still looking at carbon 1, but we see that that hydroxyl group is pointing backwards. And so this allows or causes adjacent molecules to kind of be flipped, which leads to a very linear molecule. And we're going to see that this is going to allow for a lot of hydrogen bonding to occur. And this then allows for this to be more of a structural type of molecule. So I'm going to write structure here. And because we have this flipping business going on, so where adjacent molecules are kind of flipped upside down, we're not going to be able to do the branching. So we're not going to see any branching when we're dealing with beta glucose monomers. Right, so again, just a minor change is causing a big impact on how they're able to form larger molecules, which in turn impacts how they're going to function. So this slide shows us the three different polysaccharides that we just looked at. And so over here is starch. And we have two different versions here of amylose and amylopectin. Both of these are forms of starch. One of them tends not to branch very much, whereas amylopectin does. So these would be found in different types of plants. Glycogen is the storage molecule for animals. And so you can see here there's a lot more branching going on than what we see over here in the starch. But both of these are going to be used for storage of sugar molecules. So this is how we store all of these little tiny glucose molecules inside of our bodies. And then finally over here on the right, we have cellulose. And so you can see this structure is very different than starch and glycogen. And these solid lines that are forming in here are actually hydrogen bonds. All right, so they're not covalent bonds as we typically see these solid lines. So I wanted to point that out. This is from a different textbook and they used slightly different notation here. But these are hydrogen bonds. And so that because there's strength in numbers, even though hydrogen bonds by themselves are pretty weak, you can see that we're going to have a lot of them present in cellulose. And so this is going to make this molecule actually very strong and allow it to provide structure to the plants. All right. We talked about on Monday how sugars tend to be hydrophilic molecules. However, when we start to build these really large polysaccharides, we're going to see that their ability to dissolve in water tends to decrease pretty steeply the bigger we get. And the reason for that is because every time that we connect them together, we're doing one of those dehydration synthesis reactions, and it's going to start losing some of those polar hydroxyl groups. And so therefore, it's losing part of its ability to be polar and interact with water. And we're going to see that's even a larger case here with the cellulose molecules. They tend to be even a little bit more hydrophobic even than the starch and the glycogen. So again, it's because we're losing some of these hydroxyl groups during those dehydration synthesis reactions. So again, just taking a quick look here at starch. So again, plants use this to store the sugar. So if they don't actually need it right away, they can store it. And that's great for us because when we eat certain plants, we actually, get this starch. So I think I mentioned potatoes are a really big source of starch in our diet. So this is a way that we can get sugars into our digestive system. Again, they're made of alpha glucose monomers. Sometimes they're going to form kind of a helical structure. And as I mentioned, amylose tends to be on-branched where amylopectin does branch. And so just a couple different varieties. So it would take two different enzymes to break these different forms of starch down. And then glycogen, again, this is the primary way we store glucose in animals. You'll see that a lot of the glycogen is stored in both the liver and in muscle cells. So the liver tends to be this nice storage spot. We talked about that this is where our bodies actually break down and work with fructose sugar in particular. Does anybody know why our muscle cells store lots of glycogen? Why would it be important for those cells to store it, particularly your skeletal muscles, probably even a little bit of your cardiac muscle? Why would we store glycogen there?
In case it's ever needed.
What was that?
I didn't realize my
oh, your mic was on. Sorry, I forgot to mute all of you. No problem.
Would it be so that our muscles and stuff are able to release energy?
Yeah, so remember, your muscles are going to use a ton of energy. And I wish we had time to talk about muscle contraction in this class so we could pull all this together. But if you do get the chance to take a physiology, you'll get to study this a lot. So yeah, our muscle cells require a lot of ATP in order to contract. And so that ATP is generated by the breakdown of glucose. And so we're going to store a lot of glucose in those muscle cells as glycogen. So a very important place to keep it. So yeah, so we're going to store it in our muscle cells. And a reason why it's stored in the liver has a large vascular system attached to it. And so it's able to release glucose into the bloodstream should our blood sugar levels drop and we need to get sugar to different parts of the body. So that's one reason why we have a bunch of it stored in the liver. So that's for transportation, and getting it to places that need it. We already know we need a lot in our muscle cells, so they're going to store a bunch for us. So again, this is easily broken down into glucose monomers for that quick energy, and it tends to be highly branched. Again, it's just doing branching more frequently than the starch, but other than that, they're pretty much identical. And then here was our cellulose. Again, this one is for structure. We have hydrogen bonds that are helping to hold these, I'll draw a line back over here, to keep these in place and provide strength. We're going to see that every other molecule is flipped. I have a better picture to show you that here in a second. And the reason for that is because now we're using those beta glucose molecules. And so the beta hydroxyl group is sticking out in the other direction. So they have to flip in order to line up. So these tend to be really hard to break down because it's really hard to get enzymes in here due to all of this hydrogen bonding and the structure that forms. So they make great structural things, but not very good for digesting. Here's a new one that we haven't talked about yet. Again, this one's very specific. So chitin is another type of structural polysaccharide, and it's found in the cell walls of fungi and in the exoskeletons of insects and crustaceans, right? So that hard shell. What's different about it is it has this nitrogen containing group. So you can see like this big green circle here contains a nitrogen. It also is using, it looks like I forgot to put that on here, a beta glucose molecule. So you can see in this diagram down here, here at this carbon 1, we see that that hydroxyl group was pointing towards the back. And then if we look at this second glucose molecule, we can see that it's been flipped. That oxygen is now over here in front. So it had to flip it so that it could actually form this glycosidic linkage. All right, so this is that flipping. So you see every other one is kind of flipped. And so this helps create that really linear strand. So this is how cellulose looks as well. It just doesn't have these nitrogen groups. And then here is the last one. Again, we don't spend too much time on this here in Gen Bio, but if you go on to microbiology, this becomes really important. So I do want to mention it. This is called peptidoglycan, sorry. And we've got two root words here. So glyc is referring to our sugar glucose. And peptido here is actually referring to a peptide, which should hopefully trigger in your minds a protein. So this particular polysaccharide that's found in the cell walls of bacteria actually contains both proteins and sugar molecules. So you can see here, this little blue ball is representing some of those I think sugar groups, and then we have, or the nitrogen containing group, my bad. And then the green is where we're looking at some amino acid that's attached to it. And so if we look at this drawing over here on the right, you can see that when this particular polysaccharide kind of forms its linear strands, it's actually forming peptide bonds between the adjacent ones. So in here where these green things are, these are the amino acids, and they're actually forming a peptide bond. So this is a lot stronger than what we saw with the cellulose or the chitin, because these aren't hydrogen bonds. These are actual covalent bonds, right, that are forming between the amino acids. So this forms a very, very strong cell wall for our bacterial cells. All right, and I would encourage you to look, I'm trying to see if I have, okay. In your textbook, there's a really nice one-page spread that contains all of these different types of polysaccharides that I just went over and summarizes it very cleanly. It has this picture in it, so if you see something like this, that's what I'm talking about. Great resource to help you study for this particular unit. So what do all these carbohydrates do? So we're going to see that carbohydrates have a lot of different functions within the cells. So the main thing is this storage of chemical energy, right? So I put that in a different color. But some of the other things that they're able to do is they start out as these little precursors that help us to build larger molecules. So we've seen this in the building of our amino acids already, and we'll see it even more clearly when we start looking at how we form DNA and RNA, even ATP. So forming nucleotides, we'll see carbohydrates are a part of that. We saw that they can provide a nice fibrous type of component to give structure to different parts of cells. And they're also going to indicate some cell identity. So again, if I just draw like a really basic cell, remember they can have carbohydrates that are sticking out on the sides. In some of these, I like to put little flags. So maybe that particular carbohydrate is identifying this cell as belonging to me. And so other cells will notice that, particularly my immune cells, and will say, okay, this cell belongs to Sarah, so I'm not going to destroy it, right? So all of the cells in our body have these different kinds of tags on them. Some of them are proteins, some of them we're going to see are carbohydrates. And you'll get into great depth on that when you take cell biology. So for now, we're just going to say, yes, they are there, they're important, and we'll talk about them as we work through some of our other topics. Our main focus for this particular time in the course is really focusing on how they're storing chemical energy. So that's where we're really going to spend our time. So again, we talked about this. I'm not going to spend a whole lot of time repeating this, but just a reminder that it's the beta-1,4. So again, your beta glucose molecules that are responsible for giving us the structural types of carbohydrates. And I mentioned this on Monday, but I wanted to stress it again. Most organisms do not contain enzymes that are capable of breaking this chemical bond. So we don't have enzymes that can usually break down the beta-1,4 glycosidic linkages. The other thing is it's also really difficult to reach these different bonds because of the way that the molecule basically, structures itself in those linear strands and kind of layers on itself. It's really hard for the enzymes to get in there. And also, they're much more hydrophobic. And so it's really hard to get water in there for the hydrolysis process. So remember, to break things down, we have to basically insert water into the chemical bond, and it's really difficult to do that. So we tend to not use beta glucose molecules and their larger polysaccharides for energy. Right, they're mostly for structure. However, we do consume some of them in our diet, and they are important. So those of you who are in our nutrition program here at App State, you've probably looked at this or will look at this. This is called dietary fiber in our diet. So when you look at your food labels, you'll see that there, and it is important for our digestive health. So it does help us process some other things, even though we can't break it down exactly. So just a quick mention here again about the cell identity. So again, it's used to display information on the outer surface of our cells. And they can come in a couple different varieties. So glycoproteins, as its name suggests, is going to be a carbohydrate attached to a protein. So if you remember, we had our phospholipid membrane. I'll draw a really simple one. Did someone have a question? No. Okay. So if this is our phospholipid membrane, remember that we have proteins in there that are, let me see if I can find another color. So we had like proteins that can span this whole thing, right? And then attached to this, I can put some of these sugar molecules. So this would be a glycoprotein. Sometimes we'll see that we can have glycolipids, which means that maybe I have a sugar, and I'm just using zigzags for now. to represent the sugar, that this sugar can attach here to the lipid directly. So these are the different ways that we can kind of put these display messages on the surface of our cells. And so again, it helps cells to recognize each other, recognize cells as belonging to that particular organism, that specific individual. And it can also be used for cells to communicate with each other. So the cells talk to each other. It helps with growth and all kinds of things. So these particular carbohydrates are going to be really important as you start to learn how cells kind of get along in their environments. And so here's some better artwork than my picture. So here's the protein with the sugars. They tend to be relatively small sugars, so we refer to them as those oligosaccharides. So again, oligo meaning small or little, and saccharide meaning sugar. So this would be an example of an oligosaccharide. And then over here you have sugar attached right there to the phospholipid head. So note how all of these glycoproteins and glycolipids are on the outside surface of the cell. So they're being used to help in identification and communication primarily. All right. We're getting there, folks. I see the word photosynthesis, where we're getting to the good stuff. All right, so carbohydrates, hopefully you see from this presentation, are extremely important, right? They're our main source of energy for most living organisms. And so what we're gonna do is we're gonna talk about, well, how is it made, right? And so photosynthesis is the process where plants have this awesome ability to take energy from sunlight, right, and store it into the chemical bonds that make up our carbohydrates, right? So they're able to take carbon dioxide and form sugar with it. And so this is a really, really important process. And I think it was, let's see, who was it? Was it Olivia? You and I were talking about this, that this process, right, isn't necessarily spontaneous. We have to put a lot of energy, right? You can't just go from carbon dioxide, mixing it with water and get sugar, right? That doesn't happen. Otherwise, sugar would be falling out of the sky all the time, right? Because there's carbon dioxide in our atmosphere and lots of moisture in the air. And we don't see, that this doesn't just happen, out of thin air. So photosynthesis is a really important process to help us convert this inorganic gas basically into this high energy molecule known as a carbohydrate. So this is where we're going to move to next in our lecture. I think I'm going to skip the think, pair, share just for the sake of time. But I would encourage you all, maybe I'll throw this up. Actually, I'll throw this up in our Ask It basket. You guys have been doing a great job posting stuff in there. I loved the drawings that some of you posted for the other unit that we did. But let's take I'm going to put this there, and I want you to think about the amount of energy that's stored in carbon dioxide versus a carbohydrate versus a fatty acid, and rank them based on which one you think contains the most and why. So we're going to throw that in the Ask It basket after class today, and we'll get some answers generated there. So we talked a little bit, we're going to talk more about enzymes as we go through. So I guess the important thing I want to point out on this slide for you all is we're looking at, again, our alpha glucose molecules that formed the alpha glycosidic linkages, so glycogen and starch. The enzyme that's going to help break down glycogen is called phosphorylase. And you can see that this is found in most animal cells. And it's what we're going to use. It's the enzyme that can break glycogen down into individual glucose molecules. So very, very important enzyme for us to have. Starch is going to be hydrolyzed or broken down by an enzyme known as amylase. So if you remember, we named starch amylopectin, and we had amylose. So we had those two different starch molecules. So this is the enzyme, right? Ends in ASE that breaks down our amylopectin type of things. This enzyme is actually found in two different places in our digestive system. So you have one version of it in your mouth and another version in your small intestine. So if you, I don't know, you might have done this little experiment in high school or middle school where you just take like a small piece of bread, right? That's like all carbohydrate, and you put it on your tongue and it will dissolve just based on the activation of the amylase enzyme in your saliva. So that's one way you can, put it into practice. You can, actually do something with this one at home and maybe help you remember, okay, amylase is in my mouth and it's breaking down these carbohydrates. So it helps us break down the starch. So we mentioned that energy is stored in glucose and that we're going to use that then to make ATP. So this is basically the reverse reaction of photosynthesis, right? So photosynthesis had carbon dioxide, water, and sunlight as the energy source, and it made sugar and produced oxygen. What we're going to do after we study photosynthesis is we're going to look at how we then break down the sugar to extract the energy out of it. And so this is known as cellular respiration because we do have the oxygen present here. So we will spend a good deal of time working through this as well. And we're going to use that ATP to drive a lot of those endergonic reactions. So those positive, right, delta Gs. So we'll remember that. We don't want to forget it, right? So these are the non-spontaneous reactions that require a huge input of energy. We're going to use ATP to do that for us. And if anyone's interested in knowing just how much energy is in a glucose molecule, I have a YouTube video here that you can watch on your own time, where basically they release all of the energy inside of a gummy bear all at once. And so that will be your little prelude into why we have to do cellular respiration in a bunch of little steps. Because if it happened all at once, we would look like that poor little gummy bear. So I always like to show that so that students have an idea of why it's important that when we do this process, we take our time. Otherwise, we give off way too much heat and it would not be good for us. All right, that was fast. Any questions on that, or are your brains just kind of swimming in all of that wonderful information? I don't see anything really popping up in the chat. But if you do have questions that come up, feel free to post them there, post them in the Ask It basket. I will get back to you and make sure that we get those questions answered. So now that we're super excited to learn about how carbohydrates are made, let's go ahead and take a look at it. All right. So again, today's password is photon. What things are looking like this week? Like I said, I'm probably going to continue lecturing on photosynthesis on Friday. There's no way we can do it in the remaining, 20 minutes that we have left in class today. So I will finish or continue with photosynthesis on Friday for you all. Again, trying to limit the amount of recorded videos that you have to watch outside of class. And then I just wanted to remind you about homework that you have due on Friday, and that we do have a quiz on just the vocabulary terms that are listed on your learning guide for photosynthesis this weekend. All right, so lots of learning objectives with photosynthesis. All right, so this is one of our big processes, all right, that we're gonna, try to really wrap our heads around this semester. We will look at the structure of the chloroplast briefly this morning or this afternoon. This is where all the magic happens. There are some pre-recorded lectures for you that talk about the different types of pigments that are found in plants. We're primarily going to focus on the chlorophyll as we walk through today's lecture. I'm hoping to get through a bit of the light reactions today. So those are the ones that are directly dependent on the photons coming from the sun. And then we'll have the Kelvin cycle, which I may need to discuss a bit on Monday. So we'll see how well we do through this. All right, so these learning objectives are posted on your learning guide. So again, as you're kind of working through chapter 10 this weekend, start writing out the answers, to these things, and bring those questions to class, or let me know before class if there's parts of this, that you're really getting stuck on, so we can focus on it. All right, this is just our overall flow. And so I'm just going to jump right in. All right. So what we see here is obviously photosynthesis is really important basically for all life. So you need to thank some trees today, right? Because if it wasn't for them, we would really be in poor shape. So it's really important that photosynthesis takes place, because as you can see in this diagram, everything else is really depending on it. Even if it doesn't directly eat the plant, it's eating things that do. So this is a huge cycle, like the cycle of life. And it's important for us to understand the different components, mainly so that we, are vested in protecting it too. All right, so photosynthesis is a big fancy word that basically explains the conversion of electromagnetic energy, if we were to get really specific, right, to chemical energy in the form of a carbohydrate. This reaction does require sunlight, carbon dioxide, and water, which I'm sure you all are fairly familiar with that part. And as we are going to see, it produces oxygen as a byproduct. And that oxygen is obviously really important for other living organisms, right? So we need that oxygen in order to, what we say, breathe. And I'm going to show you next week how that oxygen that we breathe is actually for our cells. So our cells are actually the ones that need it. And so very, very important process. This is one I haven't made you guys memorize very much this semester as far as chemical structures and formulas. This one, whoops, I got rid of my pen. You will want to definitely, let's just start it here. You're going to want to know this general overall equation. All right, so star this, write it down, put it on a Post-it note, like on your bathroom mirror. You can look at it every day, right, till it makes sense to you. Oh no, she brought back redox reactions. Yes, I did. Basically, the reason I'm showing you this diagram again is to show you how we're going from carbon dioxide, which is a very low energy molecule. There's not a lot of chemical energy stored in the bonds of carbon dioxide, right? You don't go to the store and buy carbon dioxide energy drink, right? It comes in your Mountain Dew or whatever, but it has the carbonation, but this isn't the energy source, right? So we don't ingest carbon dioxide for energy because it doesn't really have a lot. But once it goes through this process and turns into a carbohydrate, we're going to see that these electrons, rather than being hogged by oxygen here, are going to be more equally shared, right? So if I just draw a couple little examples here, we would see that in 3 of these 4 bonds, right, that those electrons are going to be shared equally, meaning that carbon has gained those electrons and therefore now has a richer, more chemical potential energy rather stored in these chemical bonds, right? The water molecule gets transferred over here into oxygen. And because oxygen is the same atom, right, they're going to share these equally as well. And so here, though, the oxygen has lost the electrons. So this has been oxidized. All right, so I just wanted to show you how we've seen this diagram before, but kind of following where these electrons are going and why going from carbon dioxide to this carbohydrate is so important for supplying energy for living organisms. All right, so it all relates back to chemical structure, unfortunately. Sorry for those of you who just really don't like chemistry. It just relates back. This diagram here is a nice simple diagram that I would encourage you maybe to draw in your notebooks. What we see happening here is the basic process of photosynthesis, including both the light-dependent reactions and what are often referred to down here as the light-independent reactions. So in the light-dependent reactions, we have basically, this is representing one of our chloroplasts, this green box, and we have water that's coming in, and we have sunlight that is coming in. And again, this is just in the light-dependent reaction, and oxygen that is given off. The byproducts of this light-dependent reaction are forms of chemical energy, right? So we're making ATP and NADPH. Now, NADPH may be a new molecule for some of you. If you didn't watch the energetic coupling pre-recorded lecture, it mentioned molecules like this. So I'll give you just a real brief synopsis here. NADPH or NADP plus that you see over here is a molecule that is able to accept high energy electrons and is able to transfer them to another place in the cell or to another reaction where those high energy electrons can be used to power something else. I like to think of NAD, so we've got NADH, NADP plus, and FAD that are all these types of high energy electron carrying molecules. I like to think of these as debit cards, and that the electrons that we're putting on there as the money that we're putting on, so we can load up our NADP plus with a couple of electrons, and then I can use that to spend those electrons somewhere else. All right, so it's kind of working like a debit card, in that I can charge it full of, my currency and then spend it somewhere else. ADP and ATP, I think of as cash, right? I can just directly use this energy to do something. So these are high energy molecules that are actually able to perform the work. So these are going to be really, really important for us to be able now to convert carbon dioxide into sugar. So again, we've converted this sunlight, this electromagnetic energy, into ATP and NADPH, which we can then use that chemical energy to help us convert carbon dioxide into sugar. So again, a nice simple diagram. You can talk yourself through it on these basic steps, and that will help you kind of understand as we go through this, the process a bit more. Any questions on this so far? No? All right, everybody looks good. All right. So again, our organelle of focus for this particular unit is going to be your chloroplasts. They're colored green because they contain those lovely chlorophyll pigments that are green. And this is where we're doing all of these light reactions. So the term photo refers to the light reaction. We do have a little bit of some anatomy here, right? One thing really cool about a chloroplast is that it actually contains a double membrane. So it's not just a single membrane like we see on our cells. This is an actual double membrane of phospholipids, all right, that is here. So it's, got 2 layers of protection. We see these little discs. Each disc is referred to as a thylakoid. And the thylakoids then contain the actual pigments like chlorophyll, xanthophyll, and some others, okay? Most of them are chlorophyll, which are reflecting the green light, and that's why we color them green. When we stack up the thylakoids, that's called a grana. And then the fluid that is basically surrounding all of this inside here is just known as the stroma. All right. And so we're going to look at different parts of photosynthesis that take part within the thylakoid, as well as some of it that's going to take place out here in the stroma. The synthesis part of the term photosynthesis is specifically referring to what is known as the Kelvin cycle. And this is taking place, like I said, like in this fluidy region known as the stroma. The Kelvin cycle is where we are able to take the carbon dioxide and basically take it from that inorganic molecule and turn it into a sugar. One thing that confuses students a lot, and we'll get to this probably on Monday, is that we're not directly making glucose in the process of photosynthesis. What we're making is this 3-carbon sugar known as G3P. So G3P is going to be an important molecule for you to kind of remember as we work through both photosynthesis as well as cellular respiration. So G3P is actually the carbohydrate that we make in photosynthesis, and then it goes on after that to be further modified into the 6-carbon sugar that we know as glucose. All right, so I don't want you to get confused or lost. I wanted to point that out. Our actual product, direct product coming out of photosynthesis is going to be the G3P molecule. This diagram, I believe I might have taken from another textbook just because it was nice and simple, showing us, again, these light-dependent reactions and these what they call light-independent reactions. So again, showing us that this is taking place within the thylakoid of our chloroplast and that the Kelvin cycle is here in the stroma. It's a nice diagram to show you what the inputs are and what the outputs are. So again, a nice figure, I think, for you to use as you're studying this process and kind of talking through what's happening in photosynthesis. All right, so you guys ready for a little bit more difficult concepts here? We've got about 5 or so minutes, so I will introduce the term photosystem. This is section 10.3 in your textbook. I would highly recommend that you do spend a little time reading through it and trying to answer these 4 learning objectives as you do that. So looking at how these excited electrons get transferred through the different photosystems. So the first one is actually photosystem 2, and this is where we're going to actually make ATP. The second set of excited electrons are actually going to go through photosystem 1, and they're going to produce our little credit card and ADPH, okay, our little debit card there. We will briefly talk about what is referred to as the Z scheme, and this is really just a way to illustrate how photosystem 2 and photosystem 1 are interconnected. And then again, following this flow of electrons between the different photosystems, we're going to look at something known as an electron transport chain and how this helps to increase our supply of ATP. So a lot of stuff here. It gets a little bit technical, but I'm confident that you all are able to handle this, all right? So let's take it, one step at a time. So again, the location where we are is in the thylakoid of the chloroplast. And what we're going to see inside those thylakoids are these very organized units known as photosystems. And within these photosystems, this is where we house the different pigments. So like I mentioned, chlorophyll is the main pigment. It is able to absorb the highest energy for us. But there are also some accessory pigments. So I mentioned things like xanthophyll and carotenoids. So those are the yellows and oranges you see in the fall, like when the leaves start to change color. Those are those accessory pigments. And so they are able to pick up some additional energy that the chlorophyll can't on its own. So it's just another way for the plant to maximize how much energy it can absorb from the photons of light energy from the sun. We're going to see that these photosystems, there's two of them, work together during the light reactions. And students hate this, and I will admit I do too. They're numbered backwards. So photosystem 2 happens before photosystem 1. And the reason that they're numbered backwards is because photosystem 1 was discovered first. And so it got to keep 1 with its name, and then photosystem 2 was discovered after it. So it's just confused generations of biology students, right? So I'm sorry, if I could fix it, I would for you, but it is one of these annoying things that, I don't know, I guess they thought it was clearer to leave it that way, but photosystem 2 happens before photosystem 1. All right, so everything you learned about math and the order of numbers, throw it out the window, right? So we'll get to that in a little bit. So what we're going to see is that each of these photosystems has a unique set of proteins that forms what they refer to as an electron transport chain. And so we're able to move these electrons kind of from one to another. And it's going to allow us actually to form ATP molecules. So it's a really cool process. So I will definitely get into it and geek out about it on Friday. So you don't want to miss that. So we're going to talk about how that works. And the good thing is that we use the electron transport chain in cellular respiration as well. So if you don't fully get it this week, you'll get it again next week, right? So this at least is a process that we get to hear about a couple times. Within our, what do I want to call it? Our thylakoids, we have these areas known as reaction centers, where there are these very specialized chlorophyll A molecules that are able to do a little bit more than your typical chlorophyll. And so we're going to see that these are going to help us capture these very excited electrons and kind of send them through this electron transport chain. So not all chlorophyll are created equal. And so these specialized ones are the ones that are going to help us with getting our excited electrons to the places where they need to go. So I think I'm actually going to stop there for today. I think I have definitely bombarded your brains with a lot of information. So we will pick up, like I said, with this on Friday, and we'll just keep working through this. Again, I do encourage you to, start doing the readings and work on the homework. And that way, if you have any questions while I'm talking about this material on Friday, we can always pause and I can explain it, because you can tell there's already some strange things going on here in photosynthesis, right? I mean, the numbers are already backwards. What the heck? So take a look at it. Even just review, what we did today. Get familiar with some of the terms like NADPH. What is that? It'll help you, I think, get more out of our lecture time together on Friday. So I'm going to wrap this up for today. You guys have been great. It was a quiet day. I didn't have a lot going on in the chat. So either you're totally confused or everything was crystal clear. I'm not sure which, but I'm always around to help out. So I'll stick around a few minutes. And if you have any questions, and if not, you are welcome to go enjoy the sunshine today if you're here in Boone. So have a good one. Billy, it looks like you're someplace nice and sunny.
I'm actually on Chapel Hill's campus today.
Nice.
Yeah, so I'm enjoying that. It's pretty nice.
Very nice.
Yeah, thank you so much. Have a good one.
You too. All right, let's see. Any questions here from Brittany, Bryce, or Naira? Oh, I missed Connor's question.
Yeah, I did see that, but I wasn't sure how to answer it. And I think I had noticed it after I got done, like sweeping and cleaning my room. So I couldn't tell also if we had gone over that yet or we're going to get to it.
Right, I'll put it in the chat, in the discussion forum. Just gonna go there right now and paste his question so I don't forget it. I thought it was an attendance question,
Oh, okay.
which is why I hadn't clicked on it. All right.
Oh, but yeah, you got to the part where the photo systems are named